I know we are all more interested today in the doing together the question page, which we will, right after a couple of more slides, not the whole presentation, but a couple more slides from the chapter that we're still dealing with, which is molecular genetic techniques, whatever, something that have to do with changing the genome of the organism or sequence it or stuff like that. Let's see if the pointer works. Okay, so we did together, we said a couple of things about mutations and how we can use them to induce mutations to delineate the pathway, cascades of molecules, to decide that the relation between different molecules to have a shared pathway. And then we talked about DNA cloning. So, and we finished last time, we ended up having a bacteria, a clone, that express a single DNA fragment. And now we can do a couple of things with it. And we only will mention a couple of things that have to do with characterization of the DNA fragment that we have produced. And just a word about the field that's called genomics. And we will leave out the last point. We mentioned it actually a couple of times along the course, a couple of methods that we can use to up or down regulate the gene, but we will not do it as a full discussion. That's a bonus. It's a bonus track, as I said last time. I don't know. Okay, so after we have this plasmid or this vector having inside it a DNA fragment of interest, this is the desired DNA fragment, the one we want to use or to characterize and so on. We would use usually a gel electrophoresis to separate these two by size, and then we would be able to achieve the isolated, purified DNA fragment, cloned DNA fragment. And well, there are a couple of methods to sequence the DNA fragments today. There's an avalanche of different methods the last 2 or 3 years, and new methods, very exciting ones. This is why maybe you sometimes read in popular science magazines or even in magazines the people talk about revolution in the sequence field and so on. So that's because of the so many very cheap and effective methods we have. But the traditional one to sequence a DNA molecule uses D-deoxyribonucleoside 3-phosphate. So a reminder, the nucleotides that are the naturally occurring nucleotides in the DNA molecule, the endogenous DNA molecule, is the deoxyribonucleoside phosphate. You see it right here, deoxyribonucleoside phosphate. And then the base could be a GT and so on. And what we use in this method for the traditional method for sequencing is the D-deoxyribonucleoside, meaning that we have less hydroxyl in position 3. It's not in the name. So the naturally occurring has the hydroxyl right here in position 3, linked to carbon 3. And you have just this plain hydrogen in the D-deoxyribonucleotide. These are derivatives of this one. And these are the one we use in the sequencing method I'm about to elaborate about.
Are they occurring naturally?
Yeah, I'm not sure. Yeah, that's a good question. I'm not sure. What we do know is that these molecules, I guess they're not, because of the following fact. Because this nucleotide, the D-deoxyribonucleoside, it has the ability to link to a previous nucleotide on a DNA fragment during DNA replication, but once it is incorporated, it is not able to link to what's supposed to be the next nucleotide in the chain. So the DNA replication stops with this derivative. So this one, when they incorporate it, they can link to the next nucleoside and so on. But this one, once it is entered into the DNA fragment, that's where the DNA fragment stops. Its elongation stops. Okay, so that's written also right here. So I would assume that if these would naturally, maybe at the end, I don't know, but I just don't know.
So you buy them like they are?
Yeah, they are synthesized. It's synthetic molecules. You can buy it.
The DNA copy and the copy is terminated in the natural processes too?
Yeah, but I don't know if anyone ever reported seeing these molecules in nature. That's what the only thing I can comment about. Okay, so we understand what the DNTP means, and we understand that the DNTP is the naturally occurring molecules. Okay, so now what we do in order to sequence the full fully a DNA fragment, we used 4 test tubes. In each one, we would have the DNTPs, the naturally occurring, the whole 4, G, T, C, and A. And in each of these test tubes, we will have a low concentration compared to the naturally occurring DNTPs, a low concentration of one of the DNTPs, of the DDNTPs, sorry. So this one will have a DDATP, and the second one will have DDGTP, and so on. Again, in the background, you have the whole 4 naturally occurring DNTP nucleotides, in this one as well as in this one, and in the all 4. Okay, so you understand what you have in each test tube, right? And now you allow, where you add, under certain condition, you increase very substantially the effective efficiency of DNA replication in these test tubes. You play with the temperature, with certain enzymes, and so on, and then the DNA starts to replicate. Okay, so let's focus on the second test tube, the yellow one, the DDGTP, the one that has the DDGTP. So the DDGTP, because we use low concentration, will not be the first G to be inserted, incorporated into the DNA fragment. Will usually even not be the second one, and so on. But at certain points along the DNA, there are the DNA molecules that are being replicated, this DDGTP will be incorporated. As we said, this is where this DNA fragment would stop being replicated, and you would get this product. And another possible product is this one, and then this one. And actually, this one will also be one of the products, right? Because they are randomly incorporated into the DNA molecules that are replicated. Now you take these products and you run them on a gel that has a single nucleotide resolution, meaning that 2 DNA fragments with 2 different lengths that differ between one another in a single nucleotide lengthwise, they will be positioned in 2 different spots along the gel. They will be nearby, but you will be able to differentiate between these 2. Okay, so you run these products on a gel. You run the same products of the next or the rest 3 test tubes, again on a gel. Then you align, and for each gel you would achieve this one of the colors of these graphs. Then you align all these graphs together, and you have to see a single peak in each position, right? If you have aligned it correctly, and if you have used enough DNA quantity, which is exactly what the clone DNA allows us to do, to achieve enough of the DNA molecule to sequence it very reliably. So you have to see a single peak in each position. It can't be otherwise. And so what we're looking at right now, actually, it's the sequence, right? The color that has the biggest peak, or the only peak around this region, belongs to the color, belongs to the nucleotide that we know came from the right test tube. So this is what we call, this is the most, as much as I know, the most traditional way to sequence a DNA fragment. It's effective until a certain size of a DNA fragment, and not too long a size would still work properly, but it has its limits. To be short, it has its limits. But did you understand the mechanism? That's the main thing here.
Can you repeat what the y-axis is?
The y-axis is just it's arbitrary units of okay, that's a good point. Also, you have to mark radio label. You have to radio label each of the low concentration DDNTPs. Here, the ATP and GTP node with the radio label. You have to put it well, you can use fluorescent, I guess, dyes also, but on small molecules, radio label, which be more effective, I think. The main thing is that these products that are marked in the end, because this is where they stops being elongated, they have the radio label, the radio label right here, and then this would allow you to see them on the gel. That's that. I don't know if you can do it with fluorescent dyes. I don't think so. All right. So that was. And let's just say the name for this method, because it has termination reaction. Wait 1 second. Okay, so the whole method, the whole mechanism I just described is traditionally called D-deoxy chain termination method. Just link it or give yourself a title for this method. Okay, we'll skip the movie. so we could get to the question as fast as possible. Now, okay, so we have characterized the DNA fragment fully on the sense of we got its whole sequence. Now, what we can do with it? Okay, so one thing we can do with it, we can use it as a probe to determine if there is a specific DNA fragment in some DNA mixture that we've extracted from a certain organism, for instance. So what we call the assay, that's called the Southern blotting, enables detection of a specific DNA sequence from a mixture of DNA. You start with a mixture of DNA that you've extracted from certain brain regions, say, and then usually again you start with electrophoresis, separation by size. In order to use the probe, you have to transfer the molecules from the gel to a blot, to a nitrocellulose, most usually, and you do that by applying a vertical electrical force that's similar to what we talked about in the protein case, in Western blot. Then you get a blot on the nitrocellulose, and you can then use, the radio-labeled cloned DNA fragment which will hybridize with the DNA that is complemented to it from the mixture, and if indeed you have such a sequence in your mixture, then you will get a signal. And if not, that you will not get a signal. And even if you have more or less, under certain experimental conditions, you can even quantify how high or low is this signal. So Southern blotting is the method is the version that detects a DNA sequence, and it's called after the inventor, Southern something. And the Northern blotting, is the RNA version of the same assay actually, really following the same protocol. It's just a word game. We say word game in English, game of words, on the name of the inventor of Southern blotting. So that's Southern and Northern blotting. And in each one you use the radio-labeled clone DNA fragment. Okay, another thing you can do with the clone DNA fragment is you can insert it into what we call expression systems. And these can be bacterial or animal expression systems in order to produce large amounts of protein, the desired protein. So for instance, the growth hormone that's given for children with problems of development, problems of growth. It's called sometimes growth hormone or growth factor. It's the same thing. It's protein, and it is isolated from a bacterial expression system that was inserted with a clone DNA fragment that codes for this protein. And this is again, it's a cynical use of nature, but it's very effective, very cheap. You don't have to synthesize the protein. You have the cells that do it for you. And then all you have to do is to isolate it, and from bacteria it's really easy. Now there's another example that at least I am aware of. Well, sometimes if you have a protein that undergoes some post-transcriptional, post-translational modifications, so these modifications are very different from bacterial cells to animal cells, specifically to mammal cells, for instance. And if you want to produce this kind of protein properly, you want it to function properly, you have to switch to an animal expression system. And so insulin is one example. I'm not sure from which animal. I think people today isolate insulin from a cloned animal. Is it pig? I don't remember. Okay.
It used to be pig.
Used to be pig.
Some fungi or bacteria.
But maybe they managed to do it in bacteria already today. But the traditional, maybe it was a decade ago already, they have used some mammal. I think pig. Anyway, you sometimes have to use bacterial, sometimes animal, but the expression system again allows you to produce very cheaply, more effective than any technion could ever do. So people still use it, and we use it in the lab to get large amount of protein. So I, for instance, use a cloned DNA stable line of cells that express the amyloid beta. And then I can isolate the amyloid beta, that's the toxic agents in Alzheimer's disease from the paper. And then we can use the amyloid beta for different assays, different toxic assays or whatever. So it's more cheap and actually more physiologically relevant than using the synthetic amyloid beta. Okay. So we talked about sequencing of a specific genomic region with say a gene. Now, the very drastic upscaling of the same principle I just mentioned, but again, do recall that there are different methods for sequencing today, but principally, as I said, a very dramatic upscaling of the same principle of sequencing was what gave rise to the field of genomics, which are genome-wide analysis of the structure of genes and comparing them between different species. And today, as you heard, obviously, about maybe the most heroic project, the Human Genome Project, but that's not the one. We have today genomes of at least 50 organisms that I know of. I have 50 organisms, full sequence genomes on my disk on key, and you can download it and you can do with it anything you want. And I guess there is more than 50 today, but people sequence every Monday and Tuesday, and you can get full genomes very rapidly, very quickly. So what can we do with all this information? With all this information? Pretty sure I turned it on. Okay, so this is a nice story, I think, that illustrates what can we do with such a vast amount of information. So the story goes like this: NF1. It's a gene, and it was found that a specific mutation in this gene caused a very, well, I shouldn't say nasty, but it is pretty nasty, a very bad disease called neurofibromatosis, more familiar by the name the elephant man syndrome, because of the bumps that protrude from the peripheral nervous system. That's the major symptom of the disease. Okay, and then, using this kind of information, we have discovered that, people have discovered that a region in NF1 protein has high degree of homology, is very similar to a yeast protein, yeast shmarim, to a yeast protein called IRA. And from the yeast field, we know that IRA is a gap. It's a GTPase accelerating protein. We met this fellow in the past, and we know that it affects the monomeric G protein in the yeast. And then that that pushed researchers to check the gap ability of NF1, and indeed they found that NF1 is a gap on the monomeric G protein RAS. Remember, RAS is a subfamily of G protein, of monomeric G proteins. Okay, so NF1 was discovered to be indeed a gap. And that's actually what caused it. It's a gap, and it's a gap on RAS, as I said, and RAS can induce hyperproliferation of neurons, and that's exactly what happened in the disease. The hyperproliferating neurons in the peripheral nervous system is what you see. That's exactly the bumps. But let's stop here. I just want to say a word about the homology finding itself. Okay, so what we are looking at here is the outcome of an algorithm named BLAST. The homology between these two proteins, NF1 and IRA, is on the level of the amino acids. Yellow marked amino acids are identical. Yeah, so if you have identical amino acids, you mark it with a yellow marker. And blue dot indicates that these two amino acids have a very similar biochemical characteristics. Okay, so this is how a very, very high homology between two proteins look like, just that you will get a feeling between a human and a yeast. And just one more note is that, BLAST looks for local alignments, local complementarities between two sequences, either amino acid sequences or nucleotides. And that's the reason why you see sometimes these red dots here, or purple, is it? Whatever. These dots here that indicate that the IRA in this case has some fragment here that exists in the IRA but absent from the NF1. So you have some hanging fragment here, and you have to skip this fragment to continue talk about alignment some region nearby, but not exactly of the same length. So this is what we call local alignments between two sequences, and that's the main thing that the BLAST algorithm gives you.
So is it considered as something very similar?
Yeah, it looks, yeah, definitely. You can put a p-value on this, and you can permute these sequences. You can check what is the basal complementarity. Take two sequences between a yeast and a genome. randomly chosen sequences of the same length and the same quantity of amino acids, for instance, just shuffled, and ask what would be the degree of similarity. And we'll get a much lower, about 5% of this similarity or homology.
Do you know in which range this is? Is it like 50 or 70 or how much is it like?
But you don't have only to talk, you can't really talk only about percentage. You have to always compare it to you ask what's the p-value here, for instance. I mean, how out of the ordinary is this homology compared to and randomly alignment between a human and beast? Is that what you ask? So I don't know the precise number, but this is the way you should quantify. How surprised should I be looking at this homology? So again, you can produce a p-value. There are certain ways to do it. You can, there are assumptions you can make, and then you just use a specific statistical test. What I do usually is shuffle around the sequences, and you have to check for the Basel complementarity, because if you check for human and a chimpanzee or odds, your chances to get it right, so to speak, are much higher, and just by chance. So you have to compare this homology to the Basel homology between two species. Well, then, you have methods to do it, but obviously you can't just, this is just for illustration, you can't just look at it and say homology or they are not homology. You have to put the number on that. We will skip this slide with your permission, and just one more about that. Yes?
I just push your microphone.
It's still, so it's the battery, so it's not, that's good news actually, because it means it's not my fault. Okay, so one more word, and that actually would be slide before the last. Okay, so now that we have full genomes of organisms, we can even try to identify novel genes, unknown genes. And I would just, mention the principles of how people do it. Okay, so let's look, for instance, on this bacteria. So you can think of a scanning algorithm. You can, design an algorithm that will scan the genome, and that will ask, maybe, we'll look for a starting, for a transcription start site. We have a bunch of these sequences that indicate the start site of the gene, so that will be the start point of the gene. Then look for a region of about maybe 100, maybe 1,000. You need to check with what organism you work with. So again, look for a frame of a certain number of nucleotides that would be able to code for a amino acid. Will be able to code for a protein. That's what we call an open reading frame. A sequence that you can read in triplets, because the genetic code is in triplets that we've learned. So you have to look that indeed from the beginning to end you can read it in triplets, and that it has a meaning in the amino acid domain. Okay, so if you design, and then look for a transcription end site, again you can use known sequences, consensus sequences for stop sites. Okay, if you design such a very simplified algorithm, in the case of bacteria, you'll be able to identify about 90% of the genes, and with very low amount of false positives. And it was checked and verified, and it works pretty much clean in the case of bacteria. But we learned that everyone has, almost all around nature we have introns and exons, but it's more, the presence of introns is much more robust in higher animals such as Drosophila, the fruit fly, and humans. So these genes, as you recall, have introns inside them. And these are regions that do not code for a protein. So the algorithm in these organisms have to work in a much more fragmentized manner, and then be able to say that these 4 different introns, for instance, do belong to the same gene. And this is a much more complicated task. And as much as I know, the algorithms here perform very badly, actually. And this is just to give you a perspective. This kind of algorithm is what is the tool that researchers came up with the number. Humans have 25,000 genes, something between 20 and 30,000 genes in their genomes, even though for about 5,000 to 10,000 of them, we still don't know if they have an RNA product. We still don't know if they code for a protein in nature. We just know that they look like a gene as much as we can tell. So again, just to give you a perspective of how these people came up with these numbers, well, they came up with these numbers with algorithms that are sometimes in doubt. So that's the truth. Just be aware. And just one more word, what really people do and how do they optimize these algorithms? Well, each year we learn new stuff about the consensus sequences or the consensus structure of introns and exons, and these algorithms obviously had to use all we know about these consensus sequences, all we know about, and some of them we've mentioned, talking about splicing, but that's not enough. That's what the literature tells you today. At least that's what the data shows. Okay.
But we also learn sometimes sequences an intron in a certain kind of tissue and an exon in another kind, right?
Right, but we're talking about the genome, and the genome is the same for all cells, for all tissues. If you scan for a novel genome, I mean, this information is the same for all cells, for the retina, in your toe, and so on.
Yeah, but is it the same sequences that are introns and the same that are exons everywhere?
Okay. So, well, yeah, that's true. Well, the canonical identification of an intron and an exon do not always hold, and sometimes indeed something that looks genomically wise, like an intron, remains in the RNA molecule. We just mentioned it verbally when we talked about writing. Remains, and then we still call it intron, even though it's an exon effectively right now. We still call it an intron, but we call this phenomenon intron retention, remaining, in opposed to removal of the intron from the mature RNA molecule. That's true. But still, for 90, we say 8%, this is hand waving here. So for very high percent of the cases, you can differentiate between an intron and an exon. And the alternative splicing game is played on the level of different exons that are taken out and are joined together in the mature. Most cases of alternative splicing are those of exon skipping. So you can take this gene, transcribe it with this exon or without it, with this exon and this exon, or without them both, and so on. This is usually the combinatorial game you can play with exon and introns, usually.
What does it mean encoded to the right and to the left?
Yeah, well, you have two strands of each DNA. Well, the DNA is a double strand, and each strand actually codes for genes. You have genes on both sides. So the right and left just denotes that we're looking at the two, but because they are aligned, these two strands, so you can talk about a specific genomic location, but then you have to say if you're on the plus strand or the minus strand. So an address for a gene would tell you on what chromosome you have to look at, and what position, what nucleotide position, say position 3420, and then you have to say if you're on the right side of the street or the left side of the street.
Is that what above or below the line means?
Yeah, that's exactly. So this gene would be on the plus strand, I guess, and this will be the minus. These are opposite strands. All right, so let's finish with some philosophy. Okay, so as you know, in neurobiology, as in general biology, people usually conduct experiments on model animals, and then they argue arguments on humans. Either, it can be when they study a neurological disease, it can be when they study formation of semantic memory. People will conduct experiments on mice, or rats, or whatever, flies sometimes, and then would say something about biology, for memory formation in biology, or specifically even for primates. And researchers do it with some scientific basis. In the following paragraph, it's taken from the textbook, from the Lodish textbook, tries or illustrates the scientific basis that allows researchers to make this approach. Okay, so I will read it through. Humans and mice are sufficiently related to have most genes in common. However, largely non-functional DNA sequences, such as intergenic, between genes, regions, and introns, non-coding regions inside a gene, will tend not under strong selective pressure, meaning evolutionary selective pressure. Thus, corresponding segments of the human and mouse genome that exhibit high sequence similarity are likely to be functional coding regions. Okay, so the argument is that the regions that are not similar between a human and a mouse are not functional. And so if we have succeeded to induce a phenotype in the mouse that resembles some human condition, for instance, amyloid beta aggregation, for instance, the deficit in learning a new task, or what have you, then it is most probably that the underlying mechanism, the molecular underlying mechanism, would probably be the same, because we have more or less the same genome, more than less. We have really 99%, you sometimes hear these percentages for how similar you are to a mushroom and so on. But it's very frustrating for researchers to think that the non-functional, the non-coding regions, and today we have lots of data to support this, as the intergenic region and the introns do play a role in what we are, in way of splicing, in way of gene regulation, in way of different mechanisms. And more data is being accumulated or are being accumulated for again, to support this notion. Obviously, I don't try to lishmot hakarka, to take off the ground or the basis for using animal models in cellular biology. This is what I do, also, obviously. But when you hear, a scientist tells you about, again, a mouse condition, and then just draw a line between this one and a phenomenon, post-traumatic stress disorder in humans and so on, you have to take it with a salt of grain, with a grain of salt. Because, again, the assumption that's down there, even though no one says it, the assumption is that cell biology is cell biology in all organisms. And this is more an axiom than an assumption. No one has really bothered to prove this or to give a number to how true it is. And sometimes it's not really true. And people, and I will end it right here, that in the case of Alzheimer's disease, which is the topic of my PhD study, people indeed specifically show that some differences in the genomes between mice and humans underline the troubles we have in underline or at least in part the reasons for why we can rescue or we can cure the mice, and still translation to clinical studies have not been successful yet. And you can show that at least some of it are probably due to differences in the genomes, which are, again, they are functional. That's what people discover more and more. I mentioned here just two genomic or bioinformatic tools, and you can look it up after the class. Ensemble is a genome browser. You can choose your favorite organism, your favorite gene, and just look at it and ask questions about its sequence, its homology to other species, and so on. And Struggle, it's an alternative splicing browser. It does the same for alternative splicing. This tool was developed in Gil Ast laboratory from Tel Aviv University. So if anyone's interested, you can look these two tools after class. Okay, and that actually ends up all our chapters. And we'll take maybe a 10 to 15 minutes break, and then we'll go over the question page together. Okay, so let's take a break. מתעלמים מהחלק שהוא לא קודינג, אנשים יותר מחשבים למשל, שזה גם נקודה מעניינת. אנחנו אומרים שעל הלא קודינג ריג'ן אין שום לחץ אבולוציוני. ככה זו הנחה, זאת אקסיומה. אם זה נכון, אז אנחנו מסוגלים לחשב, אז יש הסתברות הרי קבועה של מוטציות לאורך האזורים האלו, שאין לנו שום לחץ, הסתברות ניטרלית. ואז אנחנו מסוגלים לחשב לפני כמה שנים הסתכלנו משימפנזה, 3 מיליון שנה. המספר הזה מגיע מזה שאתה צריך להניח שהאזור שהוא לא מגודל משתנה באופן רנדומי, ואתה יכול לחשב את הקצב השתנות הרנדומי. ואז תגיד מתי אתה והשימפנזה מתלכדים, אז אני עושה בינוני, וזהו, בקיצור, זה כזה. בגלל זה דרך אגב, בין חדשים, יש דברים שדומים 3 מיליון שנה פיצוני שימפנזה, ויש כאלה שדומים 20 מיליון שנה פיצוני שימפנזה.
אתה לא יודע כמה זה רנדומי.
כי אתה צריך להניח הנחות בדיוק. אם אתה מניח שזה קצת פחות רנדומי, לכיוון חיובי, לכיוון שני, אז אתה כבר אתה יכול להכפיל את האסטימציה שלך ב100 גם. כן. רגוע, אתה מבין?
לא, כאילו זה משהו ממש...
רגוע במובן הלחוץ. לא, מה זאת אומרת? אני מתחיל לבדוק רק שכולם יבדקו לי, אני אבדוק.
לא, המעבדת דם שעשינו בשבוע השני בכל זאת קיבלנו.
אולי זה אמור להיות, אולי זה נמצא איפשהו באיזשהו...
לא, דיברתי איתו.
באמת?
שאלתי אותו.
ההפעה שבוע שעבר הייתה.
קריין אותה
אז הוא לא אליו, אני אגיד לו. זה חשב לי.
והיום זה שהורדת את זה אומר שזה לא עובד?
לא, זה אומר ששומעים חלש.
כן?
זה אומר ששומעים חלש.
אמרת שכששומעים חלש אפשר...
אני הצלחתי, בלי מיקרופון הצלחתי לשמוע.
כן?
זה עם אוזניות טובות.
באמת?
אז הצלחתי לשמוע. לא כל כך טוב. ואני אחליף בטריות עכשיו. לא, שיהיה אולי מישהו לשאלות אם נרצה.
תוכל לשאול אותו לגבי...
כן. זהו, זה לוקח לי כבר שבוע, אני אגיד לו עכשיו
כן, זהו, שיחתלת. יופי, תודה. There is no word for fadiha. It's an important word. Yeah.
Okay, then you have to use it whenever it fits, and then you will say, I'll understand.
Like if you're talking about someone and saying bad things and maybe look behind you, maybe there. That's fadiha.
That's a big fadiha.
מה, הוא ישמור את זה בכל השיעור או שגם יסכם? זה נראה שהוא לא מסכם אותם. לא שזה משנה, כי אני לא יודעת מה יהיה. מה נדע?
הוא אמר, אי פעם וזה מפורש שהשאלות לא נלקחו מהמאגר, או שהם יהיו בתוך המאגר?
לא, הוא אמר בתקופה שהם פתחו באיזשהו...
זאת אומרת שאם אני...
אם אתה עובר למאגר,
המאגר הזה שאנחנו עושים עכשיו?
זהו, ייתכן, הוא בוחר שאלות נבחרות בתוך המאגר, לא בוחר שזה כל המאגר.
כאילו, המאגר זה השאלות שעשינו בתוך כל זה, נכון? מעניין.
השאלה אם יש שם יותר שאלות בתוך כל פרק, והוא נותן לנו רק שאלות נבחרות משם?
או שעכשיו הוא עובר על הכל, כי אז זה יהיה ממש נבחר.
לא, כי אם כן זה גם מעט מאוד, זה שכאילו שעתיים אתה יכול רק לשאול את השאלות.
לא הייתי בכלל את זה. כן, אני לא זוכר את זה.
אז זו התקווה שלי, אבל נראה.
אני בלחץ מה אני חייב להגיד, שבוע הבא.
ועוד שבוע?
ב-21, אז שבוע הבא, שיעור הבא זה השבוע.
יום שני הבא?
כן.
אוקיי.
כאילו עכשיו, אם זה המאגר והוא עונה על זה, אז אני אהיה רגוע. אם זה חלק מהמאגר וזה, אז אני אהיה רגוע. אם הוא ישאל שאלות דומות, זה כבר יש לי. אם כאילו הוא יתבסס על השאלות האלה, אבל ימציא שאלות אחרות, ברוח הדברים, אז זה כבר בעיה. גם זה לא איזה...
האמת שיש הרבה דברים גם, דבר כזה.
כן,
צריך בכל זאת עבודה. אז זהו, צריכה לשקול את זה.
כן, השאלה אם אתה באמת עושה את זה.
רגע, מה, זהו? אפשר לקבל. יש סיכוי כזה. אבל אם הוא עבד עד לפני כמה זמן, הוא יכול לעשות. לא יודע שיש הנחיה שלילה פה.
יכול להיות.
תגיד, המאגר הזה זה... המאגר מלא, פשוט בחרתם ככה זה אורך השנה או כל פעם בסוף כל פרק כל שיעור אמור...
לא, אני חושב שזה רק כל... אני חושב שהוא מוציא חימום אבל. את אפשר לסגור, לא משנה, מה שבא לכם. וואלה.
אוקיי. אז זה לא... הוא לא לוקח הוא לא לוקח מהמאגר שפתרת בתחילת ההכנה.
כן, זה מה. כן. טוב, אני אגיד הכל. אני מתכוון. Okay, so the exam would be right here in this room, in this class, Monday next week, 8 a.m. Right? Okay, the structure of the exam is actually really exactly what you're looking at right now. So this is an example of, maybe 60 or 70% of these questions. This specific collection is from last year's exam, and then I took out some questions and put some other different questions, but generally it looks really just the same as this one. So you have a couple of questions, about 5 or 6 for each chapter, and you are told on which chapter you are asked about. And then these questions in American style, what we call multiple choice questions, usually more than 4 options, but it's multiple choice options. Sometimes you have to arrange or match numbers to letters and so on, but it's still closed questions all the way through, and then you would have something between 26 to 28 questions the whole exam, and and that's it. It will be 2 hours as the duration of the class itself, and if anyone would need any more than that, so we would switch to another room. We'd find ourselves another room and continue with the exam. All in all, it's 2 hours because there's the calculation of 2 points. 2 hours. Never mind. It's 2 hours basically, and we can, if anyone needs more time, I don't think it would be a problem. Okay, and that's about the general structure. And in the beginning of the exam, you'd have a paragraph saying how points each question is, right? What's the scoring of the principle in the exam, and exactly how to address questions that are in which you are asked a question or where you have to match things. And you have one paragraph of instructions. So I just took it out from this one. Okay, so let's start to go over the questions, and maybe for it not to be just saying the correct answer, if there is any unclear point, please do ask me, and maybe we will even open the necessary presentation or some page on the web or whatever. So if there's anything unclear, please stop me. I wanted us to do it in the second room where you can sit in a circle, so it will be a more intimate settings, but so try to be cooperative as possible. Okay, so the first question, and I also think it will be quite boring if I will read all the questions, so maybe we will take turns. Is that okay? So let's start with Nimrod. Do you mind? Yeah, so the first one.
It is customary by putting in first structural level, specify the following structures and interactions by the level in which they are found. 1, alpha Alex, 2, peptide bond, 3...
Okay, let's skip this one and go straight. So the question is clear. You have a list of structures, right, and interactions, and then you have to match each one to the structural level. It's actually a question that lots of people had troubles with it, but I didn't take it out because all in all I think it's correct. So let's start with primary structure. Okay, what of these structures you think substructures and interactions are part or defined as part of the primary structure? 2 is correct. 8 is what? It's only 2 here. So 8 hydrogen bond between the carbonyl oxygen and the amino hydrogen. So this is the bond that stabilizes beta sheets and alpha helixes also, actually, right? Hydrogen bond between the carbonyl oxygen and the amino hydrogen. So this is 8. It's not part of the program. Okay, secondary structure. Right, alpha helix, beta sheet. Right, U-turn. So these 3 are really the classical secondary structures. And also 8, as we said, because this is the interaction that underlines these structures, alpha helix and beta strand or beta sheet. Yeah. What about 4? Interactions between the residual groups. Okay, so the residual groups is really where the interaction between residual groups is really considered as a tertiary structure already.
So one alpha helix may be connected to another alpha helix.
That's a tertiary structure. Yeah. Per se, we say that a alpha helix is a secondary structure. Is anything more up level than this one or more complicated? Okay. So tertiary structure, we said already. So what do you think about tertiary structure? 4, as we said. 6, right? Globular domain. And 7 is also correct. Fibrous domain. So these are 2 domains that are considered a tertiary structure. And the quaternary structure, we are left with only one number, 9, global set of interactions between the alpha and beta subunits in hemoglobin. Never mind which protein it is. You are just being told that the global interaction between 2 domains or 2 subunits of a protein. This is what we call a quaternary structure. This one, alpha and beta so this is a subunit. A subunit in a protein means that it's a peptide of its own. Each subunit is a separate peptide, a separate small protein. Okay, so the alpha and beta here are confusing you. This is not an alpha helix and this is not a beta sheet. This is a alpha subunit and beta subunit. I should have said, and this is a good point, I should have said here X subunit and Y subunit. It's just confusing. That's right. I didn't think about it. Yeah?
Could you just repeat the tertiary structure of the quaternary structure?
So the tertiary structure, we have addressed domains as parts of the tertiary structure. So a domain, it's a region inside a single protein that has, some defined, we look at it and we say, okay, this is globular and this is fibrous and this has a sheet structure and so on. Quaternary is the interaction between different subunits, different proteins that give rise to machineries usually, or multimeric proteins.
So hemoglobin has few proteins in it?
Right, a few subunits that build it. Okay. So this is a long question. So okay. Nissim, hello?
Atom X and Y are considered bonds. Atom
Covalently.
covalently bond. Atom X more
Electronegative, yes.
mark the context sequence.
Mark the correct sentence. Okay. So what do you think? Let's draw this. So you have to draw yourself, so you have X and Y.
It's B and C, right?
I think so. Yeah, B and C. So who has X is more electronegative. So if it's more negative, it will pull the electrons. This one will have a positive, a delta charge, this will have a positive, and then you have it. The molecule would have a net negative charge. Well, you know that it's neutral because each one is an atom by itself. So taking two atoms together is still neutral. You can answer. You have to say that B is correct. B is correct from different reasons. That C is correct because the molecule would have a net neutral charge. You can say it, but by what you are being told. And B is Y would have a positive delta charge. That's also true. You see it on the board, which gives H, B and C. Okay. Okay. An atom is an element. It has neutral charge. It's not.
You don't consider an ion to be an atom?
An ion is a charged element, I guess, whatever. But if you take two atoms, link them together, and now you're telling me that one has increased electronegativity, then this is the case. I don't say that an ion is not an.
You could take two ions and not have a net neutral charge.
Again, you have to say what you are feeling shaky about C. That's the thing here. You have to say that an ion is a charged element or a charged atom.
Right.
It's not just an atom.
But
What?
because of the electronegativity, if there's electronegativity, it has to have a polar bond. They have to have polarity.
Between them.
Yes.
Yeah.
It can't be one ion which is positive or negative.
That's true actually, because then it will be not covalent, but a salt bridge or a salt kind of link. Electrostatic interactions. That's also true. Is it always true? I'm not sure.
You have the OH minus.
Yes.
So what's the
So this is the OH that's been taken out or that's been added with an electron. It has pulled, kataf has took an electron from some other molecule.
So that can
That is not,
nature.
what?
Can
Yeah, definitely. You take a water molecule, you hydrolyze it, and usually the extra electron will stay with the hydroxyl, and the proton will stay alone.
But then you can say, you can talk about electronegativity, right?
In a hydroxyl,
Yeah.
yeah, you can talk about electronegativity, only that the net charge of the molecule will not be neutral. You can talk about electronegativity in any covalent bond. Electronegativity is again, it's a property of an atom. I mean, each atom has its capacity to pull electrons compared to other atoms. So it's a property for an atom. It's a property of an atom. And then when you link this atom with another atom, it depends. In a covalent bond. Yeah, that's true. No, not in ionic bond. Then we're talking about, no, not in ionic bond. Let's leave it at this. We should enter more deeply than that. Okay, so question 3.
A double bond in a carbon chain of a fatty acid will cause, okay, so rigidity in its region.
What do you think? Rigidity in its region? It's correct, right? So all the way through it, you have single bonds which rotate continuously. So it can be like 90 degrees or 180 degrees and so on. And usually, but all in all, it's linear. And then when you have a double bond, it will cause rigidity in its region. That's correct. Anything else there? C is correct also. Bending in its region. It causes, yeah.
It might cause straightening, depends on which.
But the double bond is no. It will always cause a kink. It will always cause a kink. Let's open just the slide that shows this in the first chapter. It says double bond. You know, well, as we said, you do what? Okay, never mind. Biological systems contain the cis double bond, and the cis double bond introduce a rigid kink in it. So the question is exactly on this slide.
But is it a trans double bond?
Well, I mean, fatty acyl chains only include cis double bonds. Biological systems in fatty acyl chains in organisms only contain cis double bond. They don't have trans in them. The trans are when you cook the oil and so on, so you get trans, but
But it is biological system.
a double bond in a carbon chain of a fatty acid will cause. Okay, maybe you have a point. Yeah, so in general, you can say that it will not necessarily bend its region, but a trans. How would a trans look like? I'm not sure. Okay, maybe you have a point here.
Maybe a trans would be a kink. It would be.
I mean, the thing is, okay, so A, we are all agreed rigidity in its region. What about C? If it will be bended or not? Well, I didn't give really enough information to answer this question. If you consider what you've said, maybe in this case I would definitely consult Mona. Yeah, I would tell her what we've been taught in class and so on, and what you understand the question. Maybe we'll drop C or the whole question. I'm not sure. Maybe you have a point here. But I mean the content of the question is clear, right? So let's try to take what we can. Okay, in a chemical equilibrium, this is short. I will read it. So in a chemical equilibrium, what describes correctly the situation of chemical equilibrium? There is no transition from products to reactants. No, this is not true. There is a transition from products to reactants, so B is correct. There is no transition from blah blah. There is a transition from blah blah. So B and D, right? So H is the correct answer. Only the rates of the transitions equalize. Okay, protein structure and function. Adal, can you read?
Interaction and stabilize beta sheet structure in proteins are hydrogen bond between adjacent parallel strands.
What you say? Okay.
And hydrogen bond between polar couples in the protein scaffold.
No, wait a second. A is no. The interaction is stabilize beta sheet structure in proteins. Beta sheet structure. Hydrogens bond between adjacent parallel strands. This is true. Yeah, sorry, this is true. My mistake. Sorry.
Hydrogen bond between polar chemical groups in the protein scaffold.
Which? What do you think?
The protein scaffold is the neutral. What is protein?
No, so the protein scaffold is meaning the amino acid, phosphodiester bond, amino acid, phosphodiester bond. This chain of amino acid is the protein scaffold. So you have hydrogen bond between chemical groups, between the carbon, for instance, the carbon and the nitrogen, that builds the scaffold, that are not part of the residual group, but that are part of the scaffold itself. The backbone, it's the same. The backbone. Scaffold and backbone are the same. Here.
But then these are covalent bonds. You have also
No,
hydrogen bond between
No, so the phosphodiester bond between two amino acids is definitely a covalent bond, but you have interactions between the carbon and the nitrogen. Let me show you. Without the, we have it here. And one second, but the question is about beta sheet. So you see these interactions between the carbon of the hydroxyl that is part of the backbone, this dashed line here, and the hydrogen that is part of the amide, 4 amino acids down the helix. This hydrogen bond is what stabilizes the alpha helix, and it's a bond. It's a hydrogen bond, not a covalent bond, between this moiety and this one. Yeah, just okay. So the beta sheet is a different one, but it's the same link.
But these are not polar. These are only the alcohols are polar.
These are the same links, only different orientations, but different. And then again, what are you saying?
And I asked, we said that these are not polar. We said that only the alcohols are polar. And it's a question, these are between polar chemical groups.
Hydrogen bonds between polar chemical groups in the protein, but they are polar because this is what gives them the ability to form the hydrogen bond. They are polar simply because of the electron. I mean, they are polar groups because for instance, the oxygen is more electronegative than the carbon, and the hydrogen is less electronegative than the nitrogen. So you have minus and plus, and they are hydrogen bonded. But that's not the essential thing here. So I lost myself actually. The interactions that stabilize beta sheet structure in proteins are so hydrogen bonds between adjacent parallel strands. This is what we see right here. This is absolutely a correct answer. So A is correct. Hydrogen bonds between polar chemical groups in the protein scaffold. This is also correct. And hydrogen bonds between residual groups of the protein. So again, no, this is incorrect. Hydrogen bonds between adjacent perpendicular strands. So they're usually parallel. They're usually parallel. You should say that D is incorrect. You have A and B, which is H.
So in this question you could have also marked only one of A and
No, because and
no, I don't mean like.
You mean what will be the
These
score?
questions are in a way that I could also mark only one answer.
And then get half the points?
So
That's
I was just confused because of E, F, G, H. Because I always thought I'm supposed to mark two of them. You know what I mean?
okay. So you would have instructions of how to do it in the beginning of the exam.
Okay.
The instruction says that just choose one question, the most correct question that considers all correct all correct sentences or whatever. You would have to choose H here. If you mark A and B on the anyway, you answer in a different answer page, like a table. So you will only insert H there. That's okay. You can mark on this page anything you want. Okay, please. Can you?
Below the protein fragment is several secondary structures. The structures are numbered from 1 to 4. Mark the answers that link the structure with their names correctly.
Okay. So you have to look at this figure and identify what structures you look at. Actually, that's about it. So what do you say? Which paragraph? Which one's the correct answer? See, let's look at it together. One is the alpha helix. This one right here. Then 2 is the U-turn. Beta sheet. That's the whole. This is what marked with this circle. The whole strands together, and each of these strands, one strand is a beta strand. This is 4. Okay. Who's next?
Following is a list of terms for protein structures: alpha helix, beta strand, domain motif. Mark the correct answer.
Okay, so up till now, this list, you don't have to use it, it's just to להכניס אתכם לאווירה. get you into the environment of the, to get you into the atmosphere of the question. Yes, it tells you to think about alpha helix, beta strand, domain, and down. I will ask you a question about these terms. So, which sentence? Right, it's A, which again argues a correct argument about these terms. A single motif can contain several beta strands. Okay, we've seen it for the zig finger motif and so on. Remember, as a motif, a protein motif is something that's in this tertiary structure, that it can hold a certain or several secondary structures in it. So a single motif can contain several beta strands, and a single domain can contain several motifs. A domain is up the hierarchy of protein structure than a motif, and it can contain several such motifs.
So in the ordinary structure, the domain?
Not really. It's still, a domain is still part of the tertiary structure, but still it's in a higher hierarchy, just simply because we have it open. So let's look at one motif and then a domain. So this is a motif. You see it holds one strand, one helix, a second helix, a turn. This is a motif. And then a protein domain, it can have the zinc, this specific, say, calcium binding, motif can be right here in the middle of the globular domain, and still, it has more regions than just the zinc, just the calcium binding motif in it. So it's in a higher hierarchy than the motif. Okay. Sorry. My bad.
During the ubiquitination process, proteins are phosphorylated several times. Proteins are marked with degradation tag several times.
Let's stop here. So A and B, which is correct? Right. They are not phosphorylated. They are being tagged with a ubiquitin. That's a molecule that tags them. Okay.
Proteins are transported to the proteasome, or proteins are transported outside of the cell.
Which one? So it's the one that C, proteins are transported to the proteasome. So all in all, you have to choose F, B, and C. Okay. Now you. In allosteric?
In allosteric mechanism of protein regulation, we will see the following phenomena.
Let's start. Let's go one by one. So A.
Change the amino acid sequence.
What you think? No chance. I mean, maybe a removal of a protein fragment or something will cause this thing, not allosteric mechanisms. Okay? B.
Change induced by bonding of the molecule.
Binding, sorry.
Hmm?
I have a typo there. Never mind.
Change induced by bonding of the molecule to a protein active. What was it say? Binding.
Binding. Can I say bonding? Bonding is something else. Should be binding. Never mind.
Binding of the molecule to a protein active site.
What do you think? Allosteric mechanism is any binding of a molecule and then induce change of a to any place other than the active site of a protein. Okay, so it's definitely not B, and it's C, because C says change induced by binding of a molecule to a protein inactive site. This is really part of the definition of allosteric mechanism. And that's the only true section. Yes, C. D is confusing, but D is also incorrect, because you will not find a change in the protein's secondary structure. I think we have actually mentioned it, very accurately in the class. You would have a change in tertiary structure and up. Because tertiary structure is the level that continuously undergo minute fluctuations structurally wise. Okay, molecular genetic mechanisms. You have just two questions here. Yes.
So in the DNA molecule.
So let's go one by one.
A is the sugar phosphate backbones are located in the inner side of the WNA.
So these are biochemical questions. You have to understand what sugar phosphate backbones mean. So what does sugar phosphate backbone mean? Well, yeah, the answer is, well, A are from the sugar phosphate backbones are from the outside of the DNA, right? And this is what B tells you. Sugar phosphate backbones are located in the outer side of the double helix, so B is correct. And let's continue with C.
And bases are located in the inner side of the double helix.
You know, they complement the picture, so that's true, and that definitely says that D is incorrect. B and C, which is G. Anyone? We've completed a round, right? Yeah.
Helical structures found in both protein and DNA mark the correct answer. Protein helix contains hydrogen bonds parallel to the helix axis.
What do you think? What everyone think? So again, you remember that that a helix is something you find in DNA and proteins. And there are certain features that differentiate between these two. So protein helix, that's what A says. Protein helix contains hydrogen bonds. The helix in proteins, hydrogen bonds parallel to the helix, to the helical axis. So maybe it will be, you have to draw it. What, Matamel?
It is parallel.
Yeah, it is parallel. So in protein, the links are between, we've just seen this picture a minute ago, so the link is something between this moiety and this, so it's parallel to this is the helix, the axis of the molecule, and in DNA, it's between, okay, this is DNA. Between bases, the Watson-Crick interactions are perpendicular to the helix, to the axis of the molecule. Okay. So A is correct, and also the DNA helix contain hydrogen bonds perpendicular to the helix axis. So that's F, A and D. Adele, نمشي هنا.
Two conclusions in phospholypids have different characteristics to maintain stabilizing interactions with water. Definition: A phospholipid tail has phospholipid head, body, and entire phospholipid. Phospholipid head. A amphipathic head, hydrophobic tail.
Okay, let's stop on it. So after you can draw yourself maybe the basic structure of the molecule and tell yourself, oh, this is the tail, this is the head. Although we did it in class actually. And let's just say which is the correct one. I don't think. Let's see. Hydrophobic head, hydrophilic tail, amphipathic body. It's B. It's B, right? So it's Madam. Amphipathy is the character that you have both hydrophilic and hydrophobic nature in the same molecule. So if this is a phospholipid, the head is hydrophilic. Let's just write. What? It's B. Hydrophobic head. Mama?
Hydrophilic head.
Yeah. Amphipathic body. And what B says? Hydrophilic. It's the opposite, sorry. Hydrophilic head, sorry. I'm sorry, yes, of course. This is phil, this is phob. Because it's polar, it's non-polar, and everything is amphipathic. Sorry, it's C. Good thing you insisted. Okay, 13. Listen.
Following is the list of cellular organelles. Match one organelle or more from the list to the function of its cells in the cell. Plasma membrane, mitochondria, Golgi, nucleus, smooth ER.
Smooth ER. Okay, let's start with there. So, processing and sorting of proteins that formed in the rough ER. Who performs it? 5 is smooth ER, and it's correct. And 3, Golgi. Again, processing and sorting of proteins that formed in the rough ER. So maybe more directly would be the smooth ER, but also remember the vesicles filled with proteins are transported from the rough ER to the Golgi apparatus, and so 3 and 5. Both of them. Okay, and B, regulation of entry and exit of molecules into or from the cell or out of the cell. Yeah, so this is 1, the plasma membrane, the gate, the barrier. C, location of lipid synthesis and detoxification. So let's start with the location of lipid synthesis. Who performs lipid synthesis? The Golgi and the smooth ER. But detoxification is a feature more attributed to the smooth ER. So it's only 5. C is only 5. It's written in the slide even, I think, the detoxification issue. Okay, D, ATP synthesis by glucose oxidation. 2, right? Mitochondria. And last one, location of ribosomal RNA synthesis. Right, the nucleolus. The nucleus that's inside the nucleus. Yes?
Concerning the entry and the exit of molecules into and from the cell, couldn't like take Golgi into it? I mean, it's not regulating, but it's like performing.
Regulation of entry and exit of molecules. Yeah, it's pretty much true because yeah, indirectly I would say you have a point. It's a tough thing to write multiple choice questions in biology, that's for sure. But it's easier to check, and it's easier to answer also. Actually, that's what I think. I mean, okay, but the regulation thing here should I don't know what would be the call here. Okay, yes, transport.
Okay. Coupling of transition of an ion down its concentration gradient with the transition of an ion against its concentration gradient is done by ion channel.
No, I agree. It's not correct.
ATP powered pumps generally no.
Generally, because you have coupling of transition of an ion down its concentration gradient with the transition of an ion up its concentration gradient, not a coupling between a chemical energy by ATP hydrolysis and which would be used to draw an ion up its gradient. Right? You have a coupling of downhill transition and uphill transition. So yes, so please continue. So A and B are incorrect.
And equators.
Right.
And this is correct.
Yeah, and also symporters. And that's another subtle point. So symporters also do the same thing. Only in symporters, both molecules are translocated to the same site. So what? They still use the downhill of one coupled with the uphill of the other. Right? Okay. 15.
The resting membrane potential in living cells is negative. What is the main cause for that?
Okay. What do you think? Which one? So let's start with which you have 3 ions in a question, and for each one you can choose if it goes inside or outside. Let's choose who's the ion that determines most the inside negative state of it's potassium. Again, because passive channels, the ones that have the biggest conductance, the largest conductance in resting states, not when you have an action potential or anything, are those of potassium. Mainly the passive channels would conduct a potassium current. And under physiological conditions, what will be the direction of the flow of potassium? Outside, because potassium is has high levels, is in high concentration inside the cell and low concentration outside, and its gradient drives it outside. So it's B.
Also problematic because not the current outside that make it negative. Negativity is because of the balance.
Because of the balance?
Yeah,
It's a potential. You have, but part of the potential is driven specifically because of this current. You have the ATP pumps that drives, sodium out, potassium in, and so on, gives you the ionic environment. So that's one. And then, although maybe I'm not sure all of a sudden. if it's really a cause and a consequence. I do think it's a cause and a consequence. If you put an electrode, you have some resistance. Never mind the electrode. You have some resistance across the membrane. The resistance is actually the inverse of the conductance. So that's what the channels are, mathematically wise. And then if you allow a current of potassium outside, you would have a. I'm not sure. All of a sudden, if this phrasing is correct, what's that? Because of the yeah, I mean the inside. No, but there's another thing. I mean, no. I am sure about this phrasing. Each ion has its own reversal potential. We haven't really talked about it here. It's the battery of the ion. So this is where the ion pushes you. And the potassium is about minus 80 millivolts. And the fact that you have lots of passive channels open for potassium drives you towards this battery. So you have to have, it's the conductance, it's partly the conductance that drives you towards this battery. It's a cause and consequence. It's a cause and consequence.
It's always the relationship between the inside and the outside.
Right.
So how can one side be more responsible than the other side?
More what?
More responsible.
No, it's not more responsible, but it's you have it's not more responsible. You have a current of potassium. On that we agree. And then this current, this conductance is what pushes you to be more close to the potassium battery. It's a cause and consequence. I should maybe find the words to say it more clearly, but it is. Maybe I will send something by email. I will.
It's a passive current.
It's a passive current. That's absolutely true.
Zero current.
Passive currents are still currents, and they can give you differences in voltage. For instance, they are not the active voltage gated, ligand gated, whatever gated channels, but they are still but they still deliver currents. Okay. Neurotransmitters are packed in secreted vesicles by who? Which are driven by what? So the neurotransmitters are packed by secreted vesicles by antiporters. These are antiporters, which are driven by proton gradient. Remember, there's the coupling between first you packed or you induce high concentration of protons inside the vesicles. You make it more acidic, and then you use this proton gradient to take out a proton and to enter a neurotransmitter. That's the mechanism of the vesicle packing.
Short question about that, because I don't know the answer to that or not. At the beginning, when there is still a high proton gradient, doesn't that damage the neurotransmitters that are transferred into the vesicle?
Because of the acidic environment, neurotransmitters are quite stable molecules. They can, they can. No, that doesn't damage them at all. They can even sustain a much more harsh environment in sense of acidic and temperatures. Usually they're quite stable molecules. Okay, G protein kappa receptors.
Different receptors which function in signal transduction.
Okay, let's go one by one.
A would always activate different molecular processes.
What do you think? No, we've said that there's a convergence between the level of the receptors at the membrane. There's a convergence and a divergence. So they will not always activate different molecular processes. They will sometimes activate the same molecular. Okay.
B can activate the same molecular process.
Obviously correct. C.
C would always be activated by a different ligand. D can be activated by the same ligand.
So from C and D, which is correct? D again. Well, maybe in biology it's a principle that everything can be. Yeah, maybe so. It's not always true though, but usually
It's also
D can be activated by same ligand. That's true, and we have actually gave examples where acetylcholine can activate nicotinic acetylcholine receptors, the muscarinic, other receptors. Okay, so we have H. Okay, who's now? Enrica? 18.
The activation level of a G protein was measured in two states, A and B, at the level of dissociation between the subunits using the FRET method. In the present protein, the alpha subunit activates the effector and was marked by blue marker. Gamma and theta subunits were marked together with a yellow marker. Blue light was transmitted as the yellow emission was monitored. What can we learn from the following results?
Okay, that's a long story. It's a long story, but I'm very proud in this question. I think it's a cool question. So, okay, so what do we have? We have gamma, we have a G protein. This is alpha, the membrane. This will be the alpha subunit, and this will be beta and gamma. And then which one is? We are actually which dye are we monitoring? The yellow, which is linked to which subunit? The alpha, right? We have the yellow. Actually, it doesn't matter because when you have a drop in the emission, then all you can say is that these two components have been separated from one another. This one goes here, or the other, or one stays stable and the other dissociates from it and flow laterally along the plasma membrane. It doesn't really matter. You would get a drop when these two subunits are getting farther apart from one another. So this is exactly the principle of the FRET method, which we discussed in class. And now we're looking at two graphs, A and B. And now for the questions. Okay, so G protein is activated faster in A. What do you think? How would you identify a faster activation in these graphs? This is before we've added the ligand that activates the cascade and causes dissociation and so on. And this is after.
Earlier drop.
What's that, Enrica?
I thought if the drop is earlier, it would be activated faster.
Right. So A is activated faster, right? A drops faster, and so A is correct. G protein activates faster in A. Is activated faster in A. And so B is incorrect. And let's jump to the correct answer. D, during maximal activation, how would you identify, where would you look for the maximal activation along these graphs? In the end point, where you have achieved saturation already. So during maximal activation, more G proteins are activated in B. So if more G proteins are activated in B, you would get lower level at the saturation stage, opposed to A, or compared to A. Okay, so F, which includes A and D.
It could mean that they just go further apart from the alpha subunit.
Yes.
It necessarily means that there are more G proteins.
During during maximum activation, more G proteins are activated. You say that if it's enough, if you have the same quantity, the same amount of G proteins, and both of them have got, well, I tell you what, physiologically it doesn't really happen. If you have the same concentration of a G protein, dissociation of an alpha subunit from the G protein is total. I mean, it just dissociates from it and then link another G protein. Physiologically, it doesn't happen. Okay, 19.
In each of the big A subunits, there are two binding sites, 6 and 3. After binding to the first site, the affinity to the second site is different. How is this phenomenon caused? B, positive cooperative binding.
Right, exactly. So you have a description of a situation, and how do we call it? It's positive cooperative binding. If it was okay, you got the picture. 20. Okay, I would read one. Following are the steps of the mechanism in which G protein acts through adenylyl cyclase by order of occurrence. So this time it's not randomized; it's by order of occurrence. Activation of G protein, following by activation of adenylyl cyclase, then increased cyclic AMP levels, they PKE activation. So you get all this information for free. In which of the transitions between these steps does amplification take place? So this time biology is not always coming. You can't always say everything is always correct. So you have amplification in some of these steps, but not all of them. So between 1 and 2, between activation of a G protein and activation of adenylene C-class, do we have an amplification? What do you think? A G protein was activated. Does a single G protein activate a single or more or a couple adenylene C-classes? A couple. So you have amplification here. A is correct. What about the transition between 2 and 3? Activation of adenylene C-class. Does adenylene C-class produces a single cyclic AMP molecule or a couple? A plenty cyclic AMP molecule. So we have amplification in this one also. But 3 to 4, increased cyclic AMP levels. Cyclic AMP binds to the pKa activation site, binding site, and it has to be linked to it throughout its activation period, the pKa. So you have no amplification from 3 to 4. So it's only A and B, which is D. Maybe, let me see. Okay, so we will not make maybe we will. No, of course we will. Okay, so following are several, just one more question I'll be reading. Following are several molecular mechanisms that allow adaptation to light in the retina. For each process, the current state is described. Right below the numbers that answer correctly the following question. As the retina spends more time in darkness, so and now the okay, so one is rhodopsin is more phosphorylated. Rhodopsin is less phosphorylated. So what do you say? In darkness, what happens? Does the rhodopsin is down more phosphorylated or less? What's that? The phosphorylation to rhodopsin, what does it cause? It causes it to be less effective, less and less effective, until the alastin joins the party. So in darkness, you are naked of any phosphorylation. So the rhodopsin is less phosphorylated. I'm just looking at the wrong page. Less and less phosphorylated. Yes. Alastin would bind more to rhodopsin. No, alastin binds more to rhodopsin in light conditions, in intense light conditions. So 4 is correct also. And 5, G protein would be located at the outer segment of rod cell. This is the extra adaptation mechanism we've described. 5 is correct. The G protein will be more exposed there in the outer segment and hence more active. So all in all, it's 2, 4, 5. Sorry?
In the test, we will have another answer that says 2, 4, and 5, or can
No, you would just have in the table you have a place you would have to write 2, 6, comma, 4, comma, 5. You would get instructions, specific instructions. It's not.
And you can get partial points?
Yeah, sometimes. In these questions, yes. In these questions, yes. Still, all questions are scored the same, have the same scoring, but yeah, these questions you can be more and or less correct. Okay, let's.
Following the physical transduction of G proteins in polypods D in an arbitrary order.
Okay.
Sort them by their order of occurrence according to sequence.
Okay, let's just. If anyone has the correct order, anyone has it? I think he has it. What's the correct order of occurrence? And let's just read it one time in the correct order of occurrence. How everything starts in the phospholipase cascade. Okay, so I would tell you. Everything starts with G, with 3. G protein activation. Afterwards, which activates the phospholipase C. So 3 follows. I would just write it. So you would have a definite answer. Six. 4. Okay, so then phospholipase C activation, which then cleave, right? It cleaves and gives the products IP3 and DAG, which then the IP3 activates IP3 dependent channels down there in the ER, following by calcium increase in the cytosol because of the calcium that was stored in the ER, which activates PKC anchoring upon the membrane. It's still not sufficiently active, but it induces it to migrate to the and then anchor upon the plasma membrane, and then it has the ability to phosphorylate target proteins. Which among the following is a known following is a known function of for HNRNP, right? The function number. So I mentioned that people research the HNRNP field and attribute different roles to these molecules, but there are a couple of things. There are 3 roles that are established already, and the question is on these 3 roles. So what do you think from this list? What do we know for sure that HNRNP does?
Regulation of splicing.
Right, which is regulation of splicing. 5 is correct. 2 is also correct. Transport of messenger RNA from the nucleus to the cytosol. We've seen it. It interacts with another protein, the messenger RNA exporter, that interacts with the NPC, the nuclear pore complex, and enables translocation to the cytosol. And 3 is also correct, and we've mentioned it as well, maintaining the linear form of RNA in the nucleus.
Which were they again?
2, 3, 5. Who wants to read?
Alternative splicing.
Yeah.
Variants of the protein of the potassium channel in auditory cells allows A, fine-tuning of the cells to a specific frequency.
Is that correct? Yes. All right.
The
B.
oscillation in different frequencies for different auditory cells.
Also correct. This one enables A. Yeah.
Can you explain that?
Yeah.
Auditory cells?
Again, the different frequency for different auditory cells. So different auditory cells, specifically hair cells, sit in different locations along the basal membrane in the cochlea, and then each one will oscillate due to the fact that mechanistically wise it stands on a specific region in the membrane which is sensitive to a specific frequency. Then you have the whole cascade of splicing of what is presumed to be partly involved with splicing, which will cause the cell to oscillate in a specific frequency with the whole potassium channel thing, which will cause a fine-tuning of the cell. We've go into some of the details along this cascade. These were just bullets.
So A and B are actually the same thing?
Well, B is what enables A. They are both correct as they are. Fine-tuning of the cell's specific frequency. This is the outcome, and it is being enabled via oscillation in different frequencies for each hair cell. A and B are both correct.
Is the variant of the potassium channel the reason for the different oscillations?
Absolutely, because each variant is sensitive to different concentration of calciums, and then the fact that it opens in lower or higher levels of calcium, that's what drives the oscillation, the intrinsic oscillation of the hair cells. Yeah, so it's absolutely the cause for it. Okay, C is.
Is the location or the...
Sorry, what's that?
It says that the location
On the plasma membrane is what is the rough tuning of the cell. And yeah, and then because it activates, yeah, and that's the thing. C is specific. Before the fine tuning, I mean, when we say fine tuning, so what's the rough tuning? The rough tuning is the fact that each hair cell would just mechanistically be sensitive because of where it sits on the plasma membrane to a specific frequency, and then if it activates a lot, it will change its splicing status inside its cytosol due to functioning of different kinases that we've mentioned, and so it will changes the state of the variants of the potassium channel, which is sensitive to different calcium concentrations, which will cause the cell to oscillate in different frequencies, which gives you a fine-tuning of the cell. Okay, so sorry, C? Yeah.
Specific variants to different auditory cells and brain regions?
Not at all. It's a confusing point from the other system that people are researching, the networking between the retina and different brain regions. And D?
Different sensitivity of the potassium channel for the concentration of calcium.
Yeah, that's correct. So this is again part of the mechanism of the fine-tuning. A, B, right? Yeah, it's just F. A, B, and D. So we have 2 more questions, and I will leave it fast so we can leave in time. Okay, microRNA and siRNA molecules differ from one another by A and C, by the fact that they can be, they are in the state of single or double strand state prior to the cleavage, prior to the cutting. And C, which is they differ between them in the kind of inhibition they induced. One induced temporal, while the other induced permanent inhibition. One just induced cleavage of the messenger RNA, and the other just, again, transient inhibition in way of taking the messenger RNA to another dormant location in the cell or something like that. So A and C. F.
So what is the protein that they are cutting? That performs the cutting?
The protein that performs the cut. So it's the same one for microRNAs and siRNAs. This is why it's drosha, an endonuclease named drosha. And I think we've mentioned dicer in class on the slide. So never mind drosha. Dicer is the endonuclease that cleaves these 2 molecules. So they are not they are not different in this sense. They have the same protein that cleaves them. For some of the genes, increased transcription is coupled to longer half-life of the messenger RNA. What is the mechanism that underlines this coupling? So I think we've only mentioned it verbally, but still it's an important point, and the correct answer is B. You have positive interaction between transcription factors down there or inside the nucleus and RNA stabilizing proteins, such as the BEBPI proteins that are in the cytosol. I have not go into the details of how they are coupled, but we did say that they are coupled positively. I mean, you have more transcription factors, which will cause a more rapid transcription, and you will have a more stabilized messenger RNA that lives longer due to BPBPI, the stabilizing messenger RNA molecules. Okay, so any more questions about the exam? Okay. Contact me with anything you like by email, if you would like to set a meeting, that will be great also. Yes.
What's the rate between the assignment and the final exam?
There's a deadline that's written in the assignment.
What's the rate?
The rate 40% and 40%, and 20% is participation in the discussion, something like this. You will get the whole scoring of each one of you by ID number by email right after the assignment. You know, well, when I have the grades for the exam, I would send it to you by email even before the final grade. You would like that, wouldn't you? So I'll do that. It will be a day after the exam. You will have the grades for it. And then I would also, send the 20% participation and so on. And then the assignment will be in the future. Okay? Okay. So see you in a week. And in between, we will meet also, I guess. You all answered the teaching evaluation survey, right? Don't just know. I mean, if anyone didn't do it, so you should feel very bad about it.